Tatjana Barthel. I'm a PhD student here at the Helmholtz-Zentrum Berlin, and I'm working in the field of structural biology. And our world is a three-dimensional world. So in our research we try to figure out the three-dimensional structure of proteins, which are the smallest units of the cell. And by knowing their appearance, we can also try to find out how they work. Proteins are basically doing everything in the cell. So we also have, of course, the DNA, which carries the genetic information. But everything that gets this kind of blueprint into action are the proteins. They take care of all the processes in the cell. For example, they break down our food, they take care that our immune system functions, they transport substances from one part to another, they take care of our skin's flexibility and stability, they're responsible for our sight and smell, and so on and so on. So they are the workhorses of our cells. And when we can understand how these proteins work, we can also understand how the organism works. So we want to find out the three-dimensional structure of proteins. This basically means that we want to find out the positions of the thousands of atoms that make up a protein, where they are in relation to each other. In order to get our three-dimensional structural information, the protein needs to form crystals. And this happens in the way that billions of proteins need to order themselves into a repetitive way. And thus we get a crystal that we can then irradiate with X-ray light. BESI2 we need for the high-intense X-ray light. BESI2 is an electron storage ring. It gives us light of a lot of different energies, and we cut out one portion of the spectrum, the high-intensity X-ray light, which we use then to irradiate our protein crystals. These protein crystals, when we irradiate them with high-intensity X-ray light, we get so-called diffraction images out of them, which are then evaluated with complex computation processes. And when we calculate our structure of the protein from these processes, we can also display them. And with the structural knowledge we have then of the protein, we can try to understand how they work and then also intervene their function. So we have to get the proteins that we work with out of some cells, and when we have them, we put them in certain solutions so that they form these protein crystals. And these protein crystals we then have to mount onto a special machine here at BESI2, where we can put it directly into the X-ray beam. This hits then a detector, which then has these diffraction images. And the diffraction images are then brought to different computational programs where they are evaluated, and in the end we can solve the protein structure and display it as a kind of model. As I said before, when we know the appearance of proteins, we can understand how they work and then also intervene in their function. This is very important, for example, what we did in our project in the spring of this year, where we investigated a protein of the SARS-CoV-2, the main protease, which is called MPRO. This is a very important protein that the virus needs to multiply. So we try to intervene there. With our structural knowledge we have of the protein, we can study different molecules that intervene in the function of it, and this hopefully we can gain an active substance or even a drug in the end. Yes, we did find something, but it's still an ongoing investigation, so I cannot talk more about it. So, in our case, we approached a lab that we already knew, that worked for several years already on coronaviruses and also now on the SARS-CoV-2. And we made a collaboration with them, so we combine our knowledge that we and they have and our facility to get a novel drug in the end. So they send us the protein, the M-Pro. We do here several experiments, as I described before, that we solve the structure with different molecules. And all this information then we passed on to the other lab again, and they continue now the work. First of all, I want to make clear again that we work here on drugs and not vaccines. So there are two entirely different approaches. You administer vaccines for healthy people to protect them from infection, and drugs you administer to patients to help them recover from a disease. We basically do the first step in the whole drug development process. So we take our target protein, we search for starting points to develop a drug, and then the other labs continue this work to get it into an active substance in the end. Basically also the whole HZB, so the Helmholtz-Zentrum Berlin, gathered all the energy we could, and we already started directly to investigate this protein that I told you before, the M-Pro. So actually we were here in a kind of shutdown period, but HZB made it possible for us to start up the machine again in a time where it usually shouldn't start up. And we could do our work here. As I said, we get the protein from another lab, so we could investigate this protein with different small molecules. And then we could give this information back to our collaboration partner that they continue working on it. So for the SARS-CoV-2 project, we now collaborate with German laboratories. But for our usual projects we do here, we collaborate with laboratories all over Europe and also beyond. For example, we also have a lab that we collaborate with in Israel. And mainly we provide them with expertise from our side and also help them in the first steps, for example, to find starting points for drug development or also solving novel structures. So usually in the pharmaceutical industry, they say it takes about 10 years from finding a target protein to an approved drug. But right now, with the coronavirus pandemic, everybody is trying to make this process go very, very fast. And the whole drug development process can be separated into several steps. The first step is the research phase, where we identify a target protein, so a protein that has an effect on a disease, and then we try to find molecules that intervene with its function. When we found these, we go into another phase, the testing phase, where we test the molecules on different properties, for example, if they are toxic, and only then we can go into the longest phase, the clinical phase, where we administer this potential drug first to healthy volunteers and then to patients. Only if we go through all these steps successfully, we can get this drug approved by the respective administration. For example, in the EU, it would be the European Medicines Agency. As I said before, usually the process takes about 10 years. So we can only hope that we can cut it short to a few years now, but it's not really clear how long it will take. So during the Berlin Science Week, we want to discuss how we can utilize our knowledge of the structural information of proteins to create novel drugs.